All right, you guys, we're on lab 1.12, population genetics, and we are on the last activity or exercise C, which is about genetic drift. Okay, so very set. I'm gonna kind of breeze through this one because we should have a lot of background on how to complete these tables now that we've done a couple. So I'm gonna breeze through it a little quickly this time. So we're talking about glacier chipmunks, okay? And we're talking about how they live in a meadow at the base of a steep mountain. A gene determines their number of stripes on their face, okay? We have 2 stripes on their face represented by the dominant allele A, which is going to be represented by my black paper clip. And then we have 3 stripes representative of our recessive allele, lowercase a, and those are going to be from my pink paper clips. Okay. It says set up my initial population by placing 50 black and 50 white beans in a bowl. Again, I'm taking this down to a 10th level rather than well, I guess it's the single level. So I'm going to do 5 pink paper clips and 5 black paper clips. Okay, I put them in my bag. It says record these numbers as a percentage of the total population in decimal form in the parent population allele frequency column. So if we look at our table, here is what we're doing. Ignore this surviving offspring for a second. I'll explain that in a minute. So we have, it says 50-50. These should be decimals, just so you know. So this should say 0.5 and 0.5. I think that's a typo. So if that's confusing, I apologize. And then here we go. We're going to do our predicted genotype number and frequency. So we do the same thing that we have done in our other charts. So in order to figure out those numbers, we get our calculator out and we do p squared for homozygous dominant. So here is our p-value, which is 0.5 squared, which is 0.25. We're going to do the same thing for heterozygous, which is going to be 2pq. So 2 times 0.5 times 0.5 will give us 0.5. 2 times 0.5 times 0.5 gives us 0.5. There it is. And then here is our q squared, which is our 0.5 value over here, squared, gives us 0.25 again. To figure out how many numbers in a population that's going to be, we are going to take the number of individuals that we have, which is 50. Again, that's based on the fact that they're telling us to do 50 black beans and 50 white beans. Each bean represents an allele. Each person, or chipmunk in this case, has 2 alleles per genotype. So that means there's 50 individuals. So in order to figure out how many individuals for each genotype, we take our frequency and we multiply times those number of individuals. We have 12.5 here. Because A is dominant, this big A is dominant, we're actually going to cut ourselves a break and round down, and we're going to round up for the recessive. So we're going to go 12 for homozygous dominant. We're going to do 0.5 times 50 gives us 25 for heterozygous. And then we're going to do 0.25 times 50 gives us 12.5. We're going to round up because the recessive alleles need some love, you know? All right, so we've already filled that in together. And then we're going to do the observed. So it already gives you here examples of the observed. So in that case, if I was pulling these out myself, my hair's going crazy back there. If I was pulling these out of the bag, okay, that's what the observed are. So it's telling me that I've pulled 14 homozygous dominant, I've pulled 4 heterozygous, and 2 homozygous recessive. But wait, those numbers don't add up to 50. Why is that? So what we're doing in the directions here is we're only accounting for 20 surviving offspring. And if you read through the directions, the reason for that is because these chipmunks, we're giving them a factor that's limiting their population. In this case, it's going to be an avalanche, I think, that comes, yeah, an avalanche that comes off the mountain slope. And so only 20 individuals survive. So that's why we're only going to pull 20 times. So if you need to think about it that way, this surviving offspring is the number of individuals you're going to have to pull genotypes for. So 14 plus 4 plus 2 gives us 20. So we have those numbers recorded because we tallied those on our own. Now we need to calculate frequency. And remember, in order to calculate frequency, all we have to do is take part over the whole. So we pulled 14 out of 20. If you put that in your calculator, 14 divided by 20 gives us 0.7. So that's where that 0.7 comes from. Then we'll do 4 divided by 20 gives us 0.2. And then 2 divided by 20 gives us 0.1. So there is our number and frequency for our observed genotype, or the ones that we pulled out of our bag. And then we have our observed allele frequency over here, which is going to basically be kind of calculated in the same manner, but we have to use the equation given to us on our table. So if we scroll down, here's the equation we're using for that, okay? So in order to calculate it, we need to follow that equation. The equation says we're going to take 2 times our homozygous dominant genotype, how many we got, so that's 14, 2 times 14, and we're going to add that to the number of heterozygous, which is 4, and we're going to divide by the total number of alleles. Remember, only 20 chipmunks survived. Each chipmunk has 2 alleles, so 20 times 2 is 40. We're going to divide by 40. And that's how we got 0.8 for the homozygous dominant allele. We're going to do the same calculation for our homozygous recessive allele. That's also representative of P for dominant and Q for recessive, if you remember. So we're going to take 2 times our homozygous recessive genotype, how many pulls we pulled those for individuals, which was 2, gives us 4. We're going to add our homozygous. Remember, we have to account for that little a in the homozygous genotype. We're going to add 4, and then we're going to divide by the total number of alleles, which we already said was 40, so that gives us 0.2. All right, now we carry these on to our next line. So we're going to put 0.8 here to start our first year, and we're going to put 0.2 to start our second year. We again only have 20 individuals that are surviving, so we're going to fill in our predicted genotype number and frequency based on the Hardy-Weinberg mathematical principles. Then you're going to actually have to tally and pull from your bag for the observed over here, and then use that mathematical equation on step 6 to find your observed allele frequency. Carry those frequencies down. Notice how our surviving offspring number is changing over here. And so that comes into play again when we do our observed genotype. Your predicted is always going to be out of 50 because again, we have 100 alleles total to start that off. And we're always going to have 100 alleles total, okay? So it tells you to keep doing that. But you're also going to have to adjust how many you have in the bowl of each allele. So you'll always have 10 if you're simplifying it down, which should be 100, but 10. So you're going to have 0.8 here for the capital A and 0.2 here for the lowercase a. So that means I'm going to change how many paper clips I have in my bag. So where I had 5 and 5, I'm now going to take those out and I'm going to do 8 and 2, right? Or if you're using the hundreds like you're supposed to, it'd be 80 and 20. Either way that you do it, as long as it makes sense to you, that's what's important, that you're keeping track of the math. So I'm going to change my paper clips, I'm going to put them back in my bag, and then I'm going to pull again. And you'll notice, or you should be able to predict, that if you have more black paper clips in the bag than you do white or pink or whatever your color is, there's going to be a huge shift in the amount of individuals with the dominant allele versus the recessive allele, okay? So hopefully that helps you understand that. If you have individual questions on that, that's fine. At the end here, it says that that's when you finish your data table, you go answer questions 5 and 6 in your lab report. Right here. Describe how genetic drift changed. Is genetic drift likely to act faster on large populations versus small? So this is the paper that you submit for that lab, okay? And it's just giving you good practice with the Hardy-Weinberg variables. All right, that's it for our 1.12 lab on population genetics. Watch these at your leisure if you need help on any of those exercises.